Okay, chapter 16, pretty short chapter. It's all about DNA replication. And for this class, we're actually going to focus on bacterial DNA replication because it's quite a bit simpler. Bacterial, that circular DNA, bacteria have less DNA, and so it's a little bit simpler. So just understand the caveat to this entire chapter is it's a little bit more complicated with eukaryotes, but we're not going to worry about eukaryotes. So the process, very broadly, right? You have a double strand of DNA, all right? There's your double strand of DNA. It's going to break apart. So the first step is that the two strands of DNA get pulled apart. And so you have them separated, like so. And then this strand is used as a template to make a second strand, right, that's complementary to it. We'll do our new strand of DNA in red. And then this strand is used as a template to make another one. Like so. All right, so you start out with a double strand of DNA, and then you end up with two double strands of DNA. Each one has one of the original strands, and then one new strand in red. So that's just, it's just a very broad overview of what happens. We'll go into the nuts and bolts of how that actually happens. But first, I'm not happy unless I've managed to shoehorn some kind of math into every chapter. So we're going to do a little bit of math, easy math. You'll remember that there are two types of bases: purines and pyrimidines. The purines are adenine and guanine. Adenine pairs with thymine. Guanine pairs with cytosine. Right? We're looking at DNA-based pairing. You always see the purines pairing with the pyrimidines. And adenine with thymine, guanine pairing with cytosine. And recall also, right, that there is a directionality. There's a directionality to every DNA strand. Oops, it's already open. So if you have one strand that goes 5 prime, A, T, geez, that's ugly. We'll try that again. A, T, G, C, and then at the other end it's a 3 prime. Then the strand that binds to this one, the complementary strand, goes 3 prime, T, A, C, G, 5 prime. So this A binds to that T, this does that. So you get complementary base pairing along the entire length. And there's a directionality to it. So one strand goes this way, and then the complementary strand goes that way. So here's a practice question for you. If you have a sample of double-stranded DNA that is 31% adenine, all right, you can write that down. You have a double strand of DNA that is 31% adenine. What percentage is cytosine? If that double strand of DNA is 31% adenine, What percentage of the double-stranded DNA is cytosine? And this is real easy to figure out. Right? Think about it this way. If it's 31% adenine, what percentage is thymine? 31%, right? Because there's an equal amount of adenine and thymine. So that means the adenine is 31%, thymine is 31%, right? Easy enough. So you know that G and C have to make up the other 38%. So that means that half of that is going to be C and half of it's going to be G. So you're looking at 19% and 19%. So 19% of that double-stranded DNA is going to be cytosine. Real simple. But to answer that kind of question, you have to understand the base pairing relationship. So you just have to put on your problem-solving hat and use what you already know to figure it out. It's pretty simple. Okay, so back to DNA replication. And again, we're talking about bacterial DNA. So we're talking about a circular strand of DNA. All right, so here's our bacterial DNA. And there's one point on this circular strand of DNA where replication begins. And that origin point is called the origin of replication. We'll say it's right there. Right? So that's where that's the origin of replication. That's where replication begins. So when replication starts, remember the first thing that has to happen is that double strand of DNA has to pull apart. So spreading out from that origin of replication, you'll see two strands. Two places where the circular DNA forks, one right here, one right here. And we call that a replication fork. One there, one there. And those replication forks will extend all the way around this entire circular strand of DNA until they reach a point over here, and then your two circular strands of DNA will break completely apart. So they'll spread until you have two new strands of DNA. So this first step, right, this step where the double strand of DNA is broken apart is facilitated by a protein called helicase. And that's not a very creative name if you think about it. It's an enzyme, right? You can look at the name of it. ACE, it's an enzyme. So it's a protein that can catalyze a reaction. In this case, the reaction that splits apart double-stranded DNA. So it's an enzyme, and it's got this prefix right here, helit, right? Because what is it doing? It's breaking apart the DNA double helix. So it's helicase, right? So this is the enzyme that breaks apart double-stranded DNA. Write that down. So yeah, it's the enzyme that breaks apart double-stranded DNA. So we're going to zoom in on one of those replication forks, and we'll take a look at all the machinery that's involved in DNA replication. So we're going to look at, let's see, I'll make my DNA black. There we go. This is the 3' end here and the 5' end. And this double strand of DNA is getting split apart into two separate strands. So here's a 3' end right here, and there's 5' end right there. Helicase is doing all the unwinding here. So we'll make helicase in red. All right? And that helicase will extend along the entire length of this double strand of DNA. And as the helicase moves along this double strand, it's splitting apart the double helix. And so you're left with the one strand down here with a 3' end, another strand up here with a 5' end. Now, in order to keep those double strands from just re-annealing to each other, right? So helicase unwinds the double helix. In order to keep those two strands from just coming back together and sticking back together, there's another protein, another series of proteins that will bind to the single strands of DNA. And those proteins are called creatively single strand binding proteins, or SSBPs. Single stranded binding proteins. And they will just bind to the single strand of DNA and keep that DNA strand from re-annealing to the other DNA strand. And so that'll happen to both of these strands. So now they're blocked. They can't stick back together. So that's what single strand binding proteins do. Okay. So this is step one, right? Step one is just pulling apart that double helix and keeping the two strands from sticking back together. And that's done by helicase and the SSBPs. So I'm going to erase this and rewrite it. And we're going to talk about the next step, which is replication. So I'm going to throw my replication fork back up on the board. And what did I put on top? I put 3' on top? Yes. Guess it doesn't matter. 3', 5'. Doesn't matter as long as I get this right. 5', 3'. So I'm not going to draw helicase and the SSBs again because this has already been pulled apart. And we'll talk about how replication happens. Okay. So, the first thing that has to happen is, well, I'll go ahead and tell you that the enzyme that polymerizes DNA can't just synthesize a new strand. It has to add on to an existing strand of nucleic acids. So, because of that, there has to be a strand already in place that can be extended. So the next step is actually the laying down of RNA primers. right? So there's a protein called primase, which will lay down an RNA primer. And RNA also has the same directionality to it, so this is the 5' end here. It'll lay down an RNA primer. And then another protein called DNA polymerase 3 will add new nucleotides to the 3' end of this open RNA strand right there. It adds new nucleotides to the 3' end of the new DNA strand. So primase will lay down an RNA primer. That's what this is. This orange is an RNA primer. And then DNA polymerase 3 will extend, adding new DNA nucleotides to the 3' end of that RNA primer. And that'll go all the way along, right? So as the replication fork extends in that direction, right, with the two DNA strands being pulled apart, right, as the DNA strands get pulled apart, DNA polymerase 3 will just extend this strand along with the replication fork. Right? Okay. So that's what happens on this strand right here. And it works out pretty well because, you know, the 3' end, remember this, DNA polymerase 3 can only add to the 3' end of a new DNA strand. That's a big deal, all right? Sear that into your memory. DNA polymerase 3 can only add new nucleotides to the 3' end of a new DNA strand. And that works out okay for this strand right here because that extension can just go along in the same direction as the replication fork. But what about this strand up here? All right. If primase lays down a new RNA primer, well, let's say we'll put an RNA primer here. All right. What's the directionality of this RNA primer? It's 3' over here, and it's 5' over here. So when DNA polymerase shows up and starts extending this RNA strand, which way is it going to extend? Away from the replication fork. So when DNA polymerase 3 extends this RNA strand, it's going to go that way. And so what happens on this strand up here is that the DNA is replicated in fragments. Right? Down here, we were able to replicate the entire thing in one go. You lay down one primer, and then that primer can be extended all the way, just following behind that replication fork. But on this strand up here, replication has to go back in the other direction. And so as this replication fork is pulling apart that double strand of DNA, the DNA is being replicated in these fragments going back in the other direction. So this will get opened up a little bit more, another primer lays down, goes back a little bit, and then it gets opened up a little bit more, and so on and so on and so on. And so these fragments that are made happen along the entire length of the piece of DNA that's being replicated. And we have a special name for these fragments. They are called Okazaki fragments. Japanese fella named Okazaki identified them, named him after himself. This is what you do. Okay. So yeah, just clarifying here, this is our new DNA, and so is this. Down here, we were able to just lay down one primer and extend it all the way, but up here, we had to lay down multiple primers and replicate the DNA in fragments, and hopefully now you see why. So what you end up with, at every replication fork, there are two strands, right? At a replication fork, you break apart the double-stranded DNA helix into two strands of DNA. One of them we call the leading strand. It's called the leading strand because it's leading the DNA replication. You know, there's no delay between when the double-stranded DNA gets pulled apart and when this strand gets elongated. But this one gets replicated in fits and starts. So it'll replicate, get pulled apart a little bit more, another primer lays down, replicates backwards, DNA gets pulled apart again, another primer lays down, and so there's a delay behind the leading strand where this strand gets replicated. It lags behind replication. So that's why we call it the lagging strand. All right. I'm going to erase some of this, but not all of it. I'm just going to declutter the board here, and I'm going to leave my DNA and my RNA primers. We're just going to declutter. So what you can do is you can hit pause, and then you can write yourself a new replication fork, and we'll pick it up when you get back. All right. So this is where we're at so far. We've got our double-stranded DNA pulled apart. We've laid down RNA primers and then extended those RNA primers using DNA polymerase III to add new DNA nucleotides onto the ends of these RNA primers. All right? But at this point, the primers are still there. We've still got RNA in here. And the Okazaki fragments are not joined. There's a fragment here, there's a fragment here, and they're not connected. So the next step is carried out by an enzyme called DNA polymerase I. DNA polymerase I replaces the RNA primers with DNA. So DNA polymerase I will pull up these RNA primers and then replace them with DNA. So we'll do that. But notice what I'm not doing. DNA polymerase 1 does not connect the Okazaki fragments. All it does is it replaces the primers. And we'll replace our primer down here. So we've pulled up the RNA primers and we've replaced them with DNA. And then the last event is carried out by another enzyme called ligase. Ligase, as the name would imply, ACE, it's an enzyme that ligates the two fragments together. It joins together or ligates the Okazaki fragments. So ligase will come in here and then just ligate those two ends together, and now you have one continuous strand of DNA on that lagging strand. And so that's it. Those are all the enzymes that you're going to learn about with DNA replication. And yeah, so back to our circular piece of bacterial DNA. We'll throw that guy back up on the board. Well, we're really only going to look at, you know, the origin of replication here. So let's say here's our double strand of DNA, and here's a replication fork, and then on the other side of the origin of replication, you have another replication fork. So there's a 5' end here, 3', and over here we've got 3', 5'. So this is our origin of replication. Origin of replication right here in the middle, roughly in the middle. All right? And so on one hand, on one side of the origin of replication, you've got the lagging strand up here and the leading strand down here. And then on the other side of the origin of replication, it's the opposite. Now this is the lagging, and this is the leading. And what's important is that you need to be able to look at a replication fork and tell which side is the leading strand and which side is the lagging strand. And the only thing that you need to remember is that DNA polymerase 3 can only add new DNA nucleotides to the 3' end of a DNA strand. If you can get that straight, then figuring out which is leading and which is lagging is easy. Because, you know, if this is the 5' end here and the 5' end is over here, right, on the new strand, this is the 3'. So this can extend all along the length. That's why it's the leading strand. And then the other one has to be done in fragments, which is why it's the lagging strand. So if I give you a replication fork with the 5' and 3' ends labeled, you need to be able to identify which is the leading and lagging strand. Recap. Step 1, helicase unwinds the DNA double helix. Step 2, the single-stranded binding proteins bind to the single-stranded DNA and just block the DNA strands from sticking back together. Step 3, primase lays down RNA primers. Step 4, DNA polymerase 3 extends the 3' DNA strand. 3' end of DNA strand. I didn't give myself much room there, did I? I'll go with 1 underline. Don't forget it. And then step 5, DNA polymerase 1 will replace the RNA primers. And then the last step, ligase. Ligase will ligate the Okazaki fragments together. So you might be watching this and following along, and you might get the impression that all of these different enzymes and proteins are working independently of each other, because I've talked about them independently of each other. But the reality is that a lot of them get lumped together into this gigantic multi-protein complex that just does everything. And it's all part of one big machine that extends along the entire replication fork. So we call that the DNA replication complex. And the components of the DNA replication complex are helicase, all right, one of them. We got one helicase protein. You got 2 DNA polymerase 3 molecules, and then a whole bunch of connecting proteins. And if you look at the, there's only one slide on this PowerPoint, and it's a bunch of videos. So you can look at, yeah, those are pretty good videos. I'm not going to watch them right now. Watch them on your own time. There's one that shows the leading strand and the lagging strand and kind of explains that a little bit more. And then that second video shows you the DNA replication complex and how all those proteins work together. Yeah, so watch those videos. It's good. They're pretty good. There's a lot of good videos on YouTube explaining all this stuff. So you know, the more different ways you can find to look at all this stuff that we're learning, I think the better, right? Because I'll show it to you one way, and then a YouTuber might show it to you a different way. Just keep working at it until it all makes sense. Come to office hours. Okay, so what do you think? Do you think DNA replication, is it endergonic or exergonic? Right? Think about this. DNA replication, you're starting with, you know, lots of individual nucleotides and then stringing them together into this massive molecule that has a lot of structure to it and a lot of organization to it. So if you guessed endergonic, you'd be correct, right? So it requires a ton of energy making DNA. So how we're going to power all this, right? It kind of presents a conundrum because, you know, DNA is big and complicated, and so it's going to take a lot of energy to make DNA. Fortunately, the building blocks of DNA have a lot of energy already stored in them. So if you look at the DNA building blocks, right, the individual nucleotides One of them is deoxy ATP. All right, you remember way back to when we learned about ATP. ATP is an RNA nucleotide. The only difference between ATP and deoxy ATP is just one little hydroxyl group, right? It's the difference between RNA and DNA. So if you're making DNA, then the building block is just deoxy ATP. And there's just as much energy stored in deoxy ATP as there is in just regular ATP. So that's one of our building blocks. And thankfully, it's already got energy stored in it because it's basically just ATP with one hydroxyl group removed. So that's actually what gets polymerized when the DNA strand is elongated. So if the DNA strand is being replicated and the next nucleotide is adenosine, that'll come from deoxy ATP. Just, you've also got deoxy TTP and deoxy GTP and deoxy CTP. And all of these have energy stored in them, right? Remember the grossly oversimplified structure of ATP, right? You have an adenosine with 3 charged phosphate groups on it, right? That's the grossly oversimplified structure of ATP. But you can see, you've got all these negative charges bound together real tightly. They're repelling each other. And so if you just clip that bond right there, then these two would just shoot off and it would release a lot of energy. And you only need one of these phosphate groups in the finished structure of ATP. So there's actually 2 phosphate groups that can be clipped off to release energy. So if you look at how they all get stuck together, you can see how every time a new nucleotide is added, that nucleotide brings all the energy that it needs to catalyze the reaction. So let's say you've got a strand of DNA that goes 5', and then you've got deoxyadenine, right? That's just an adenine. And then a phosphate group. And then deoxythymine. And that's the end of the line, right? This is a very short DNA strand. Let's say the next nucleotide is cytosine. So the building block here is deoxy-CTP. So deoxycytosine with 3 phosphate groups. Right? So what will happen is that phosphate group right there will get stuck onto the 3' end of this strand. And what you end up with is, well, we'll draw it underneath. Now you've got 5' deoxyadenine, phosphate, deoxythymine, phosphate, this phosphate right here, deoxycytosine, 3'. So the cleavage of these two phosphate groups, the release of these two phosphate groups, releases the energy that catalyzes the reaction of taking this phosphate group and sticking it onto the end of that nucleotide. So now you've basically just added a cytosine to the end of this DNA strand. So it went from being AT to ATC, right? And so DNA polymerase 3 will do this along the entire length of the DNA strand, just cleaving off phosphate groups and attaching the nucleotides to the end, like that. All right, so I'm going to give you a demonstration of one problem that can happen when that double strand of DNA is being pulled apart. So what I've got here is a rope, right? It's two strands of rope that are wound around each other. And so this represents your double strand of DNA. So I'll let you zoom out a little bit so you can see what's going to go on here. All right. In fact, I'll pull it back so you can see what's happening in my hands. All right, there's our replication fork right here. There's the replication fork. And so helicase will unwind these proteins. And I'm going to zoom out even more so you can see what happens. There we go. All right, helicase is going to unwind. All right, you see what's happening here at the end? I'll let you see over here. All right. You see what happens there? It tangles up. It actually knots up right there. See this knot right here? That's called a supercoil. And it's actually, it'll twist up like that. So this can't unwind anymore. Right? That's called supercoiling. So that problem that we just looked at called supercoiling, this is what happens when you try to unwind a double strand of DNA without doing something to relieve that tension. The tension just builds up in front of the replication fork, and then it causes that supercoiling of the double-stranded DNA. So there's actually a really good video in the PowerPoint, a YouTube link, that shows you how it gives you a little probably better view of that supercoiling, and then how it's relieved. The protein, the enzyme that relieves supercoiling is called topoisomerase. So the supercoiling that's created by that replication fork will get relieved by this enzyme topoisomerase. So it relieves the supercoiling caused by the replication fork. And if you want to watch the animation, I recommend watching that video, the third video on the PowerPoint. So now we're going to shift gears and come back to eukaryotic DNA. Remember the big difference between eukaryotic DNA and bacterial DNA? Because up until now, we've just been talking about bacterial DNA. The big difference between eukaryotic DNA and bacterial DNA is that eukaryotic DNA is linear, while bacterial DNA is circular. So, with bacterial DNA, you can follow the leading and lagging strands all the way back, and eventually they'll just, the replication forks meet on the other side of that circular DNA, on the other side, opposite the origin of replication, and the whole thing gets replicated pretty simply. But with eukaryotic DNA, if you follow that replication fork all the way to the end of the chromosome, then you'll see a problem that'll arise. So let's say here's the end of a eukaryotic chromosome, right? You could follow it all the way to the other end, but this is one end right here. All right? Remember, when the lagging strand is replicated, it's replicated always at the 3' end. So if a primer lays down right here, or even if a primer lays down all the way over here, that primer is going to be extended that way. Right? So this is our 3', 5'. This is 5' over here. All right? So RNA polymerase 3 is going to extend that DNA strand that way. And then RNA polymerase 1 is going to rip up this RNA primer. But now there's no 3' end to add a new nucleotide on. So this chromosome just got shorter. Right? It got shorter by the length of an RNA primer, which is not very long. But if you think, over however many hundreds of thousands of rounds of cell division happen in a multicellular organism, right? That shortening can add up. So every time the cell replicates, every time the DNA is replicated, it's going to get shorter. DNA, the chromosome is going to get shorter and shorter and shorter. So, the solution here that life has come up with is to create a big buffer zone at the end of a chromosome, because the end of the chromosome, every time it replicates, is going to get shorter and shorter and shorter. And if you put important genes out there at the end of the chromosome, then they can get deleted if the chromosome gets replicated enough. So there's a region at the end of either end of a DNA chromosome that's just gobbledygook. It's unimportant, there's no genes there. It's a region called a telomere. And this is just non-coding DNA at the end of a chromosome. And so it's there basically just as a buffer, because every time the DNA gets replicated, the telomere is going to shorten just a little bit more. So then that kind of brings up another interesting problem, right? And that is, when you pass on your chromosomes to your offspring, if there was no way to extend these telomeres, then the chromosome that you pass on is a little bit shorter than the one you got from your parents, and the one that they pass on is a little bit shorter than the one they got from you. And with each generation, you have a shorter and shorter and shorter chromosome, and that telomere gets shorter and shorter and shorter. But that's not actually what happens, because in the cells that actually undergo meiosis and make those gametes, they have an enzyme, they express an enzyme that can extend these telomeres. That enzyme is called telomerase, or telomerase, or whatever you want to call it, telomerase. And what telomerase does is it extends the 3' end of the telomeres so that another primer can lay down and extend it a little bit farther. And this is a gene that gets turned on in a lot of different types of cancer. So you can imagine if, you know what cancer is, just, it's basically what happens when a cell starts replicating over and over and over again when it shouldn't. And if a cell starts replicating over and over and over again, it runs the risk of running out of telomeres unless it switches on this telomerase gene, which would allow it to extend its telomere, and it could get in a lot more rounds of replication. This is also the reason why cloning full-grown animals is a little bit more challenging than what people initially anticipated. You probably don't remember this because this didn't happen in this happened before your lifetime, but you might have heard of Dolly the sheep, right? Dolly was one of the first full-grown organisms to be cloned, and Dolly was, an adult sheep when she was cloned. But her clone didn't have the extended telomeres. They didn't turn on telomerase before they cloned Dolly, and so the clone started out with the already shortened chromosomes. And so Dolly's clone didn't last very long because eventually the chromosomes started shortening to the point where important genes were getting cut up, and so the clone didn't last very long. So that's just the kind of thing that can happen if the telomeres aren't extended. And that is the end of chapter 16. So we got one more chapter, chapter 17. Stay tuned.